Hello, welcome to part 7. You have come to the end. We need to look at the second half of cell respiration, and which is going to be the citric acid cycle or the Krebs cycle. And we need to look at this idea of the electron transport chain and what does this mean for ATP. Please keep in mind that just as a refresher where we left off, um, at the end of uh, glycolysis, we have in the bank two net ATPs. We created four, but we had to pay the bank back two from the investment phase. And so we have two to the good for ATPs. And we also created these two molecules of pyruvate or pyruvic acid, for which we used to create a molecule of, or two molecules of acetyl coenzyme A. It should be noted that each of those pyruvic acids that we created at the end of glycolysis, each one will fuel or power the Krebs cycle one time. So we can actually crank the Krebs cycle twice for each molecule of glucose that enters into the process of cell respiration, starting with glycolysis. Because again, at the end of glycolysis, we have two pyruvates or two pyruvic acids. Each one of those will create a molecule of acetyl coenzyme A, which in turn will turn the Krebs cycle one full crank. So we get two cranks of the Krebs cycle for each molecule of glucose that enters into glycolysis. And we're not going to spend as much time with the Krebs cycle because it is scary. All right. But I'm going to give you the important things that you need to know, which are mainly what are the byproducts? What are we getting out of this cycle of the Krebs cycle, and what is the purpose? And I will tell you that the purpose is you end the Krebs cycle with exactly what you start with. So, in other words, we start the Krebs cycle with this thing called citric acid. You end the Krebs cycle, right, with the precursor to citric acid. What does all this mean? Well, acetyl coenzyme A is going to join with a molecule that is known as oxaloacetic acid. Right? So oxaloacetic acid joins with acetyl coenzyme A to form citric acid. And the rest of the Krebs cycle is about taking that citric acid, breaking it down, and recreating oxaloacetic acid so that it can join with another acetyl coenzyme A to form another molecule of citric acid to crank the Krebs cycle once again. All right. So what does this look like? Right? What does this look like? Well, here's the Krebs cycle. Okay. Again, I told you it was ugly. There's a lot of enzymes in here, and there's a lot of um, um, redox reactions or reduction and oxidative reactions that are taking place here. But here's what I want to draw your attention to. All right. Here is pyruvic acid being converted into acetyl coenzyme A. And look at what we produce. We produce a NADH. Right? We produce an NADH. We produce one of those nicotinamide adenine dinucleotides. And we create a molecule of CO2. Right? That is what we're creating when we ultimately produce this acetyl coenzyme A. Acetyl coenzyme A merges with oxaloacetic acid to form citric acid. Then watch. We come through. Don't worry about what all of these little intermediaries are. All right, but we're coming down, and look, we create another molecule of NADH. We create another molecule of CO2. So now that's two NADHs, two NADHs, two CO2s. Right, we're going to come along a little bit more. Oh, we're creating another NADH. And we're creating another CO2. Keep going. Hey, we're creating an ATP. Keep going. Hey, we're creating this thing called FADH2, flavin adenine dinucleotide. Flavin adenine dinucleotide. So we're creating one of these guys. And we're continuing along, and we're creating another NADH. And by the time we create this NADH, what do we have again? Oxaloacetic acid. You start and end with the same thing. So that this oxaloacetic acid can now merge with another acetyl coenzyme A to produce another molecule of citric acid to be able to run this again. Remember what I said. This happens twice. Each glucose molecule is producing 2 pyruvic acids, which is enough to produce 2 acetyl coenzyme A's, which will turn this cycle twice. Why is that important? Well, that's important because look at our end products. For each pyruvic acid that creates acetyl coenzyme A that merges with oxaloacetic acid to create citric acid, we are creating 3 carbon dioxides. That's going to be important. That's what we exhale. All right. We're going to create 4 NADHs. Here's why NADH and FADH are important. They carry hydrogen protons. They carry hydrogen protons. And that is critically important for the electron transport chain. Right? And we'll talk about that there in a few more minutes. Right? So for each crank of the Krebs cycle, we are creating 4 NADH molecules. We are creating 1 FADH2 molecule, 1 flavin adenine dinucleotide, and we're creating a lonely molecule of ATP. But remember, we're cranking this how many times? Twice. So in actuality, we are producing for each glucose molecule that enters into glycolysis, we are producing 6 molecules of CO2. We are producing 8 molecules of NADH. We are creating 2 molecules of FADH. And we are creating 2 molecules of ATP. We can add these 2 molecules to the 2 from glycolysis. And now we have 4 net ATPs produced so far. Yes, technically 6, but remember we had to pay back 2, so we're at a net of 4. And all of these guys all now head, I shouldn't say not all of these guys, the ATPs go to the ATP bank. NADH and FADH now go off to the electron transport chain. And the electron transport chain is all about the oxidation, the removal of a proton from NADH and FADH. And the H is hydrogen. It's a hydrogen proton. Remember, it's only got 1 proton in it. That's it. It is a proton. And so the electron transport chain is all about stripping away the hydrogen proton off of NADH and FADH2 so that those hydrogen protons can be used to do oxidative phosphorylation, which is the process of creating ATP through the use of a hydrogen proton gradient that was created from the oxidation or the stripping away of hydrogen from NADH and FADH2. Let that sink in. Oxidative phosphorylation is the production of ATP powered by a hydrogen proton gradient that was created from the oxidizing or the stripping away of hydrogen protons from NADH and FADH2. Here is the stripping away of the hydrogen protons. And so you can see down here you have your NADHs, your FADH2s. You've got some proteins in here that are directing the oxidation, the stripping away of that hydrogen proton. And look, accumulating on the outside of the cell membrane, or I should say the inner membrane of the mitochondria. Accumulating on the outside of the inner membrane of the mitochondria are hydrogen protons. And those hydrogen protons charge the outside of the inner membrane of the mitochondria so that it is really positive. And what happens when you get all of these positives together where they try to repel one another? So how do they do that? Well, they've got to find their way to the other side of the membrane. And the way to get to the other side of the membrane is through an enzyme called ATP synthase. ATP synthase. ATP synthase is this guy right here. All right. And so what are the components of this ATP synthase? Well, ATP synthase is made up of a couple parts. It's got a rotor, it's got a rod, and it's got a knob. And what ends up happening is the hydrogen protons that are in high concentration on the outside of the inner membrane of the mitochondria pass through this stator. Right? This stator is this it's almost like a headrest, right, by which the rotor sits on. And so the hydrogen protons come down here into the stator, and that powers the rotor to turn. And in turn, it cranks the rod. Right? And as it does that, it actually brings the hydrogen proton down into the inside of the inner membrane. And so you can see hydrogen proton comes passing through the stator. Right? That hydrogen proton cranks itself around the rotor. Right? As the rod turns, it forces the hydrogen proton out. And the rod is then turning this knob. And it's the job of the knob to add the phosphate group onto the ADP, creating ATP. You've done phosphorylation. You've added the phosphate group onto the ATP by using a hydrogen proton concentration gradient. How frickin' efficient is that? I'll tell you. Because we do this process of chemiosmosis, this idea of using a hydrogen proton concentration gradient to fuel and power an enzyme such as ATP synthase for the sole purpose of doing phosphorylation, adding the phosphate group onto the ATP, the electron transport chain produces 34 ATPs. 34 ATPs. Why? Because we're not using ATP to create ATP. See, that's the downfall of glycolysis. Glycolysis is using ATP to create ATP. Remember the investment phase. We invested 2 ATPs. We don't see that with the chemiosmosis or with the electron transport chain. Instead, we see using a hydrogen proton to create an electrical charge that will power the enzyme to create the ATP through phosphorylation. Where in the world did our body come up with such a great idea? Bacteria. This is an evolutionary remnant to when mitochondria, which is where this is taking place, was an independent living organism. Mitochondria, 3.8 billion years ago, was a bacterium that eventually formed a symbiotic relationship with other bacteria. What? So our mitochondria are bacterial, and it kept this basic principle of fueling structures using a hydrogen proton gradient. Bacteria today still use hydrogen proton gradients to fuel its little flagellum. Bacteria don't have enough ATP to be able to produce to power their flagellum to keep them moving. It's too expensive. They're too tiny. They would burn out. The amount of heat that would be dissipated from having to create that much ATP would literally singe that little bacteria. And so they use hydrogen proton gradients. And the earliest mitochondrial ancestors maintained that when mitochondria developed this symbiotic relationship with other bacterial cells bigger than them. Mitochondria doesn't have the same chromosomal structure that we do. In fact, they've got circular DNA that is completely separate and independent from every other genetic material in the human body. It doesn't do mitosis. Mitochondrial don't engage in cell division such as mitosis. Binary fission, which is the same process that most bacteria use to split into two. Your mitochondrial are bacterial. It's a remnant leftover, highly effective, primitive bacteria living in each and every cell of your body. And it takes that primitive mechanism of creating ATP and it maximizes it during the process of cell respiration to give you the biggest bang for your buck. And you take those 34 ATPs and you add it to those 2 ATPs that we created during the Krebs cycle, and you add it to the 4 ATPs that you created during glycolysis, and you create 40 ATP molecules from each molecule of glucose. But remember, you got to pay the banker. And so we got to take 2 of those ATPs away. And what do we end up with? You end up with between 36 and 38 ATP molecules. In muscle cells, they're not as effective. The mitochondria is typically not as effective. But in skeletal muscle cells, there's an overwhelming number of mitochondria. So it kind of evens out. But mitochondria and skeletal muscle produce about 36 ATPs overall. All right. Fascinating evolutionary connections that are living in us today in every cell that we rely on. We would die. We would not be able to exist as a complex multicellular organism if it wasn't for these primitive symbiotic relationships that we have formed and we still maintain today with bacteria. This is a pretty picture that shows you where glycolysis happens. Right? Glycolysis is happening within the cytosol, right? And then the Krebs cycle and good old transport, the electron transport chain is happening within the mitochondria. The picture right here, this just kind of tells you when we talk about cellular respiration, we're talking about aerobic respiration typically, right? Because you got to do something with that electron, right, from the hydrogen, and so it's usually the oxygen that's accepting the electron. Well, there's two other ways of doing types of cell respiration: anaerobic cellular respiration and fermentation. Right? And anaerobic respiration, the final electron acceptor is not oxygen. It is some kind of inorganic molecule. And because of that, it yields fewer ATP molecules. So typically, anaerobic respiration, not done in the presence of oxygen, will yield between 2 and 36 ATPs. Now, I will say this. Anaerobic respiration still engages in glycolysis, Krebs cycle, and electron transport chain. Right? And during glycolysis, it still produces a total of 4 ATPs. During Krebs cycle, it still produces 2 ATPs. Where it varies is during the electron transport chain because it's not using oxygen as that final acceptor, and so it's not as efficient at releasing hydrogen protons. And so it's not going to be as efficient. It's not going to be efficient in producing as many ATPs as it does during aerobic respiration. All right. And so it uses nitrate ions. It uses sulfate ions. It uses hydrogen sulfide. That's H2S over there. And it uses carbonate ions. That's this guy right here. Right. So this is a nitrate ion. This is a sulfate ion. This is hydrogen sulfide over here, and this is a carbonate ion right there. Then we've got this little thing called fermentation, which I absolutely love. Fermentation is amazing. Fermentation does not use any kind of, no oxygen is required. And here's the thing, fermentation does not do the Krebs cycle. Fermentation is a process that is done by bacteria and yeast, and depending on what is doing the fermenting will ultimately depend on what your byproduct is. Right? So both bacteria and yeast, many types of bacteria and yeast will do fermentation. So they still do glycolysis, right? Which is why they produce 2 ATPs, right? 2 net ATPs. It's because of glycolysis. The actual process of fermentation does not produce any ATP. All right, and so this is a highly ineffective way of doing things. But your bacteria, for the most part, will do fermentation and will produce something called lactic acid. Right? So this is what gives Swiss cheese the holes. Right? Bacteria are producing gas that's being released as this lactic acid, and that's is what curdles the milk and makes the cheese makes the milk go bad, which is what produces the cheese to begin with. Same thing with sour cream. Same things with cottage cheese, all that stuff there that is really bad, sour, old milk is all produced because of lactic acid released from the bacteria. But think about how many good things we get from that, though. Right? Think about all the cheeses that we get. Think about the yogurts that we get and all that kind of good stuff. So our food supply depends on lactic acid and fermentation. But when yeast engage in fermentation, they still produce a net of 2 ATPs through glycolysis. But it is in fermentation, the yeast do not produce lactic acid. The yeast produces ethanol. Right? Yeast produces ethanol. Um, Cervacia. All right, Cervacia. That's a type of yeast that produces. That's brewer's yeast. Right? It's what we use to produce and create milk, or I'm sorry, not milk, beer. All right. The end product of fermentation, though, done by yeast, is ethanol. It's grain alcohol. And so all of your beer, your wine, your bourbons, your whiskeys, your vodkas, all of those things are all dependent upon yeast fermenting sugar. What creates the difference is the boiling temp, the type of yeast that is used, and very often the type of grain that is used. All right, so whether it's barley, whether it's wheat, whether it's rye, whether it is some kind of mash that you use like within beer, like corn. So the grain that you are using very often determines whether or not you're going to make a bourbon or a whiskey, or whether you're going to make a vodka or a bourbon, or whether you're going to make beer, or whether or not you're going to make wine. And of course, with wine, what's being fermented is the fruit. It's the grapes, typically grapes, right, that are being fermented. And so it's not a grain at all. It's not a grain at all. Most of your beers are going to be wheat based. All right. And then your barleys and your ryes and your corn is what's going to be producing your bourbons, your whiskeys, your vodkas. So fascinating stuff for fermentation. It is horrible at producing ATP, but we get so many good beneficial things from the process of fermentation. So this is just a nice little summary table for you. This goes back to way back, video 2, 3, on looking at diffusion and osmosis. All right. Endocytosis and exocytosis is right here. So this is kind of a nice little summary table of passive and active transport that I wanted to give you guys. And so at that, you have come to the end. I'm not going to do a review of mitosis. I'll probably post some things for you for that, but I'm not going to do a video on mitosis, or I should say the cell cycle, because we covered that pretty heavily in the lab. So I will post some review stuff for you for that. So there's not going to be a video on mitosis specifically. All right. But I will give you some review material on that. And so with that said, congratulations. The 2 hardest chapters are done, in my opinion. Chapter 2, the biochem. Chapter 3, cell bio, is now done. These are the most heavy laden content chapters that we have through all of AMP1. So you have survived, in my opinion, the worst of the course. Everything from here on out is all about application and use of everything that we have learned in chapters 1, 2, and 3. And so congratulations, and I'll catch you on the flip side.